So this topic is on bacterial genetics and variation. And last time we covered the first part, that's the bacterial genetics. And we learned the composition of bacterial genetics. And we know that there are chromosomes, this is bacterial DNA, chromosomes. And also we learned there are accessory DNA, including plasmids. That's a small circular DNA, which can contribute some properties to the bacteria. And also we learned transposons. Transposons are a special DNA fragment on other DNA. It cannot replicate by itself, but it can jump on the chromosome and also jump on a plasmid. So that's a special component of bacterial genetics. We also learned something on bacteriophages. So that's a virus which can infect bacterial host. It's a special type of virus. And today we are learning the variation of bacterial genetics. Variation means change. Any kind of change, we call it variation. So we'll learn what type of changes are there, and also we'll learn the methods we can use to change the bacterial DNA or bacterial genes. And sometimes we want the microorganisms to change so that we can manipulate their genetics, we can manipulate their DNA. But sometimes we don't want them to change. For example, during this pandemic, and we sequence the virus, the coronavirus, and based on the sequence, we can design vaccines. But you don't want the virus to change their RNA so quickly, so fast, because when your vaccine is available, you want the vaccine to work, to target on the original version of the virus. You don't want the virus to change to something else. So that's the variation topic. Okay. First, we'll learn the different mechanisms of bacterial variation. Bacterial variation. The first mechanism is gene mutation. Gene mutation means that the bacterial gene changed by points or by large scale. Anything change, we call it mutation. The other topic, the other point we want to talk about is gene transfer and recombination. So this gene transfer and recombination means that the gene can go from one bacteria or one species to another. And we'll learn what is transformation, and we'll learn conjugation, and also transduction. For some of the topics, we actually touched a little bit previously. For example, conjugation. When we learn the structure of a bacterial cell, we learned the sex pili, and we mentioned a little bit that the sex pili is involved in conjugation. And also transduction. That's somehow related to what we learned last time, the bacteriophages. So that's what we'll cover today. We'll first talk about the gene mutation. Mutations in bacteria. There are a lot of types of mutations. And keep in mind that this mutation means a lot. Any form of change on the DNA can be defined as mutation. For example, base substitution. We know that the DNA sequence is due to the base sequence ATCG. So this base substitution means that one base is replaced by another. For example, originally it was A, then after mutation, the A becomes C. So this we call base substitution or deletion. For some reason, part of the DNA is gone. It might be a short fragment, maybe a long fragment, a large fragment. It's gone. This one we call deletion or insertion. Somehow, some DNA fragment from outside enters the cell, and then it integrates into the bacterial genome, insert into the previous chromosome, and that is we call insertion. Insertion of a DNA fragment into the chromosome can change a lot. It can disrupt the original gene and split the gene into two halves, and that will inactivate the gene. Or some insertion into the genome will silence the gene downstream of it. Or duplication. Duplication means that there is one copy of the gene on the chromosome, and for some reason it duplicates another copy and insert that copy into another place. This we call duplication. Or inversion. Inversion means the gene sequence changed. You have a DNA fragment in the chromosome, and then somehow the DNA fragment inverted from backward to the end to the head. So the end switched to the head. This we call the inversion. And these mutations can arise in the bacterial populations due to different reasons. The first reason is that it's spontaneous. Spontaneous, that means that naturally. So there is no inducing agent. So it's just happens. When we talk about the bacterial DNA replicate, it's somehow accurate. And when we say accurate, we mean relatively accurate. But always there is some mistake happens. For example, when the DNA replicate, there is a wrong base placed into the sequence, and that will induce one spontaneous mutation. And after a long time, the DNA will accumulate more and more mutations. So this is spontaneous. And spontaneous mutation is the drive of evolution. Another reason is induced. Induced means that some factor from the environment changed the bacterial DNA. And this procedure we call inducing. And these factors from the outside we call mutagens. Mutagens means that those factors which can make mutation on the DNA. So this we call mutagens. This includes both physical mutagens and chemical mutagens. Physical mutagens include like UV light. So when you shine UV light on the DNA will change. So this we call the physical mutagen. Another is chemical mutagen. Some chemicals, such as the chemical we mentioned previously, like glutaraldehyde, and those chemicals can make the DNA change or make mutations on the DNA. So these are the chemical mutagen. And also antibiotics. Antibiotics doesn't change the DNA directly, but antibiotics can selectively promote some mutations to be enriched. Like in the bacteria, the mutations happen all the time. When you apply antibiotics, most of the bacteria will be killed. But if there is some mutation which becomes resistant to that antibiotic, that strain or that cell will be selected out, and it will arise again, forming a lot of antibiotic-resistant bacteria. So antibiotics is a pressure, but also it's a drive to select resistant bacteria out. Okay, I'd like to show you a cartoon, and this cartoon will explain how antibiotics promote the enrichment of mutations in bacteria. Can you hear the audio? Okay, great. Let's continue.
Here's our patient. The patient has received all known antibiotic treatments to no effect. In conclusion, either he is not ill, or we have a new strain of microbes.
Or this could even just be a case of resistance to antibiotics. Let me explain. When the body is infected by bacteria, it attempts to defend itself in several ways, by increasing its temperature, but mainly by using its immune system. As soon as a foreign body enters the organism, it's detected straight away and alarm bells ring. Lots of white blood cells come along to identify the culprits and get rid of them. But sometimes these defenses are overwhelmed by bacteria. That's when the illness really begins. Luckily, since antibiotics were discovered, we can help patients protect themselves. These drugs destroy most bacteria or create a hostile environment for them.
It's getting better?
Antibiotics soon became a quick fix solution. For the slightest scratch, a course of antibiotics. It was so effective in preventing infections from developing that the use of antibiotics increased. They became widely used to treat battery-farmed poultry and pigs. But before long, the illness returns. And antibiotic treatments suddenly stop working. What happened? Well, before bacterial cells divide, they duplicate their genetic material. This material is composed of DNA. Portions of the long molecule can hold precise information on the activity and development of the bacteria. These portions are what we call genes. Each gene is composed of a sequence of 4 elements, called nucleotides, of which 2 are always matched in pairs because they're complementary. As DNA is copied very rapidly, errors in the copy frequently occur. These errors are called mutations. Most of the time, these mutations are of no consequence, but sometimes they can be harmful to the bacteria. However, with such a high number of mutations, some of these copy errors can result in resistance to antibiotics. When in contact with the antibiotic, the resistant bacteria will be the only ones to survive. In fact, the drug will have chosen the resistant strain. And then, since there is no longer any competition for food, the bacteria can multiply very fast. It's exactly like natural selection. Individuals with the most favorable characteristics reproduce more than the others and will be able to continue to evolve. Indeed, with antibiotics, we are reproducing what nature did 4 billion years ago that led to the evolution of species.
Well, miss, are you demonstrating here that the patient is suffering from acute Darwinitis?
You could say that.
Aha, that's exactly how I see it. Gentlemen, this case is very interesting. Let's move on to something else.
Yeah, so antibiotic resistance is what happening in front of our eyes. That's the evolution of bacteria in front of our eyes. With the application of antibiotics, we killed most of them, but we gave them pressure so that they evolve and they can pick up other genes. They can change their own DNA so that they can survive. They can deal with the pressure of antibiotics. So that's evolution in front of our eyes. Okay, so these are the mutations in bacteria. Then we'll talk about a second point. That's gene transfer in bacteria. Gene transfer means that, for general, means that the DNA fragment or gene from outside, no matter where it comes from, enters the bacteria and either as a plasmid to express it or integrate into its chromosome. So that's gene transfer. There are some properties or features of gene transfer. First, it's unidirectional. So this transfer is unidirectional. It's from donor to recipient or from outside to the competent cell. Unidirectional means only one direction. Second is that the donor cell does not give an entire chromosome. If the DNA is from a donor cell into a recipient cell, the donor cell just gives maybe part of its chromosome or some accessory DNA, but they don't give an entire chromosome. And next point is that gene transfer can occur between species. Between species means that some bacteria can give some DNA, for example, a plasmid, to another species which is relatively far away. For example, you can use E. coli to give a plasmid to some other bacteria you never heard of, like Francisella. And that's not limited within the species. So these are the several features of gene transfer. Okay. In this field of gene transfer, we'll learn several aspects. The first one, we'll learn what is transformation. So transformation means that the gene transfer resulting from the uptake by a recipient cell of naked DNA from a donor cell. The donor cell can give some DNA out, and the recipient cell can take up or uptake the DNA into the cell. Okay. After taking up the DNA must be expressed to change the recipient cell, to contribute some new property, and that we call it transformation. So you transform the recipient cell to a different state. There are some factors of influencing the transformation. First is the DNA size and state. DNA size and state. If the DNA is too large, it's not easy to be taken up by the recipient cell. And also the state. If the DNA is like single-strand DNA, that's not easy. If it's a double-strand, supercoiled, that's better. And also, if the DNA is from the environment, we need to also consider factors in the environment, such as DNA molecule is sensitive to nucleus including like DNAs, which can degrade DNA. And you don't want the DNA to be exposed to nucleuses. Another point is the competence of the recipient. Competence means the ability of the recipient cell to take up the DNA. And different bacteria are different because some bacteria are ready to take up the DNA, while another species, they don't like the extracellular DNA to get into their cell. So different abilities of the recipient cell. And we need to think about the competence factors and what factors can influence the competence. For example, the growth state of the recipient cell. If the recipient cell is at its log phase, that's what we learned from the growth curve. When they are fast dividing or replicating, that's a good state for the recipient cell with a high competence. And we can also use other factors, for example, chemicals, to treat the recipient cell so that we can induce the cell and make them competent. And this we call the induced competence. And in our research lab, we always do this. We culture the bacteria and we treat them with chemicals so that we can make them competent. And then we can use that competent cell to take up a plasmid, to express it or to expand it. So that's different factors influencing transformation. And in this figure, it shows us what transformation is. There are some steps. The first step is the uptake of DNA. This is the recipient cell, and it contains its own chromosomal DNA, for sure. And these are the DNA fragments from donor cells. These DNA fragments somehow are released by the donor cell. And when you mix them together, and some DNA fragment can be taken up by this recipient cell and enters the cell. And this fragment carrying gene capital A and capital B get into the recipient cell. And then the next step is that the DNA, some of the gene can integrate into the chromosome. This procedure we call the recombination. So after recombination, this capital B gene is now integrated onto the chromosome DNA. And now this gene B, capital B, can be expressed so that this transformed cell will have a new feature expressing capital B gene. And this capital B gene might be a virulent gene, might be a resistant gene, or some other features. So now this cell is different from the beginning, because at the beginning it can only express this little b. Now it can express this capital B gene. So this is transformation. And transformation is a useful technique. We can use this recombinant. We can make this or use this procedure to make recombinant DNA technology. Okay, I'll show you a short video on transformation, and it will explain these steps, maybe in a clear way, a better way, maybe.
Hi everyone, and welcome to Biology Professor. Today I want to talk about genetic recombination in bacteria. Bacteria cells are pretty cool because they have the ability to take up foreign DNA and actually incorporate it into their own genetic profile in order to make the proteins that that foreign DNA encodes for. This can give bacteria cells extra functionality. For example, they might gain new DNA that has that codes for toxins, or that codes for virulence factors, or that codes for antibiotic resistance, and the bacteria cell can actually gain new functionality through this way. There are 3 different mechanisms by which bacteria can obtain foreign DNA. These are transformation, conjugation, and transduction. I have videos on all 3 of these mechanisms, but today let's talk about the first one, transformation. Transformation is when a bacteria cell takes up foreign DNA from its environment by bringing that DNA from its extracellular environment across its plasma membrane. The ability of the cell to take up DNA across its plasma membrane in this way is called competence. Many types of bacteria are naturally competent in that they can do this process with ease. Examples of such bacteria include species of Bacillus, species of Staphylococcus, and species of Streptococcus. And of course, there are well-known human pathogens within each of these types of bacteria. So now let's look at this mechanism in a little more detail. Here we have our recipient bacteria cell. This here is its chromosomal DNA. So this is the DNA that it has had since it was produced by replication of previous bacterial cells. But there might be foreign DNA in this cell environment. I'm going to draw this foreign DNA in red. This is DNA that is just going to be floating through the cell's environment. It might, for example, be from a previous bacteria cell that died and lysed, and pieces of this donor cell's DNA are now just floating around the environment. The recipient bacteria cell can actually take this DNA across its plasma membrane so that now this DNA is inside the cell. Then, through the process of recombination, the bacteria cell has its chromosomal DNA that has recombined with this foreign DNA. So the foreign DNA has now been integrated into the chromosome. There might be some additional foreign DNA that was not integrated, and it's just going to be sort of broken down. Also, the bacterial DNA that was replaced by this foreign DNA is also going to be broken down. And now we have this genetically transformed bacteria cell. This new DNA that is taken up can give it different functions. For example, there are species of E. coli that actually produce Shiga toxin. This is a toxin that they obtain from the bacteria type Shigella. So Shigella produces Shiga toxin Some E. coli cells can produce it too because they're able to go through this genetic recombination process. This DNA could also encode for virulence factors. For example, one such virulence factor might be a capsule. If the bacteria can produce a capsule covering its outer cell wall, this may enable it to hide from a host immune system. And so this is why the process of transformation is very important to bacterial cells, and that it can enable them to survive in different environments they might not have been able to otherwise.
Okay, so it's another way to explain transformation. Okay, another method has also mentioned in the video is called conjugation. This one we talk about a little bit when we introduce the bacterial structure. And we learn that the bacteria can produce special pili or special pillars called sex pillars. So that gene transfer from a donor cell to a recipient cell by direct physical contact between bacterial cells. And the mediator in this procedure is sex pillars. Okay, so here is the schematic drawing of these two types of cells. Here is one donor cell, which carry this plasmid. We call the F plasmid or F factor, and it can produce sex pili, which is a longer pili, hollow fiber, and it's longer than the common pili. And a recipient cell, which doesn't carry a plasmid and doesn't produce the pili. And let's see what happened. So during the conjugation, this is the F plasmid, and this bacteria is the bacteria which can produce sex pili, sex pillars, and this is the donor cell. And the sex pili can grab this recipient cell. So the sex pili can touch on the surface of this recipient cell and then draw those two cells closer and closer. And when the two cells attach to each other, and on the surface of contact, there will be a channel formed. There will be a channel formed or bridge formed, so that through the channel, the donor cell can duplicate and transfer its DNA, and in this case, it's the F plasmid, into the recipient cell. And you know, the DNA transformed into the cell is single-strand DNA, and then the single-strand DNA will be a double-strand DNA will be made based on the single strand. Now you have two cells carrying the same plasmid, F plasmid, right? So both of them now are F plus, so F positive. And both of them now can express sex pili. So this is procedure we call conjugation. So this is a conjugation. Step by step, you transfer the DNA from one cell to another and change the recipient cell from F minus to F plus. So this is conjugation. Another term related to conjugation is called high-frequency recombinant strains. High-frequency recombinant strain means that this is a special situation that this F plasmid, the F factor, is now not an independent plasmid, rather than it integrates the DNA into the chromosome DNA. The bacteria carry the integrated F factor, now can transfer its chromosomal gene to recipient cells with high frequency. And those strains, those bacterial strains are called high frequency recombination, or high frequency recombinant. That's a typo here. I'll show you this in a better way in this picture. Like this one, you have a donor cell. This is the donor chromosome, and this is the F plus cell. It carries the F plasmid, the F plasmid. And for some reason, this plasmid get into the chromosome. It's integrated, inserted into the chromosome. And this strain we call HFR cell, or we call it high frequency recombinant strain. You know, it carries F factor on its chromosome. It can still express the gene on the plasmid on this fragment. So it can also produce sex pili. Now the sex pili grab another cell, the recipient cell, and pull them together to form a channel between them, so that the donor cell can transfer its DNA into the recipient cell. But here it's different from conjugation, because here there's no plasmid, so this donor cell will transfer its chromosomal DNA into the new cell. You know, the chromosome is so big, so usually it cannot transfer its whole chromosome into the recipient cell. So usually only part of its chromosome, the DNA fragment, is transferred into the recipient cell. And in this recipient cell, the incomplete F plasmid, or the DNA fragment, can somehow recombinate into the chromosome to make change on the chromosome. Then usually it's not a complete F plasmid transferred. So at this situation, this recombinant cell is still F minus. It does not carry the whole plasmid sequence. It carries only part of the plasmid sequence, but part of its gene is different from the beginning. So this we call the high frequency recombinant strain. Okay, it's similar to conjugation, but a little bit different. There's no independent or free plasmid involved. Okay, so this is another term related to conjugation. Okay, and transduction is a gene transfer mediated by bacteriophages or the virus. So that's what happens. Genes transfer from a donor cell to a recipient cell by way of bacteriophage. And we learned that bacteriophage can infect a bacterium cell. And when it infects the bacterial cell, it will carry its own genome into the bacteria. But it can also carry other genes. So that's what we are learning, transduction. There are two types of transduction. One is called generalized transduction. It means that the gene transferred is random. So any gene can be transferred by a bacteriophage. So that's why we call it generalized. The other one we call specialized. Specialized means that it can transfer only particular genes. So only some special gene can be transferred. Usually this happens when a prophage is formed. So, you know, last time we learned when the prophage is formed, the phage DNA or phage genome is inserted into the bacterial genome. So those adjacent sequence or adjacent genes of the prophage can be transferred, or the neighbor gene of the prophage can be transferred. Okay, let's watch the procedure of generalized transduction first. So generalized transduction is the transduction in which potentially any bacterial gene from the donor cell can be transferred to a recipient. So this is what happens. You have a donor cell, and there is a phage comes in. This phage can infect this donor cell. It injects its chromosome into the donor cell. And in the cell, the genome is replicated, and it can produce enzyme to degrade the host cell DNA, degrade the bacterial chromosome into pieces. And also it utilizes the cellular enzymes and the biosynthesis system to make its own material. And then new viral particles is assembled in the bacteria. So this is something interesting happening now. When the new viral particles are assembled, it packs the viral genome inside the virus particle. But sometimes it assembles the host DNA fragment, this purple one, into the viral particle by mistake. So this packing of DNA is random. So any fragment of the host DNA can be assembled into the new viral particle. And now you have this viral particle, which is special. It carries the phage shell. So it carries the phage protein, but the DNA inside is the bacterial DNA. And this phage, well, it's not a real phage, can infect other cells. For example, it can infect another cell we call recipient cell. It can inject this DNA into the recipient cell. But keep in mind, this DNA is from the donor cell, but not the phage genome, right? And this DNA can recombinate. This DNA fragment can recombinate into the recipient chromosome, like this, right? Now, the recipient cell will carry the donor DNA, like this, and replicate with it. So this procedure we call generalized transduction. So, because this packing of the virus particle can pick up any fragment, so this packing is random. So potentially any donor DNA, any donor gene can be transferred. So that's the mechanism. That's the reason why any gene can be transferred. Okay. I'd like to show you a short video on this generalized transduction. According to feedback from previous years, a lot of students prefer the videos, and they think that with the help of the videos, it will help them to understand our material. So that is why I'd like to insert some video into our file, and hopefully those videos can help you to understand what we covered just now. Generalized transduction.
In generalized transduction, a segment of DNA is carried from one bacterial cell to another by a bacterial virus called a bacteriophage or phage. The phage attaches to the bacterial cell and injects its nucleic acid into the host cell. A phage enzyme is produced that breaks down the host DNA into smaller fragments. phage DNA is replicated and phage coat proteins are produced. During formation of the mature phage particles, a few phage heads may surround fragments of bacterial DNA instead of phage DNA. The phage particle carrying the bacterial DNA infects another cell, transferring the bacterial DNA to the new cell. When the bacterial DNA is introduced into the new host cell, it can become integrated into the bacterial chromosome, thereby transferring genes to the recipient. This cell then multiplies and carries new genetic material.
Okay, it's pretty simple, right? It's generalized transduction. Okay, then we'll learn another type of transduction, which is specialized. So during this specialized transduction, there is a formation of prophage, and the transduction in which only certain donor genes can be transferred to the recipient. And this specialized transduction is mediated by lysogenic or temperate phage, which can form prophage by inserting its genome into the host chromosome. Okay, so this is what happened during the specialized transduction. Okay, you have a bacteria. Now you have a phage infect the bacteria. And it happens that this phage is a lysogenic or moderate phage. It does not lyse the bacteria directly, but it inserts its genome into the bacterial chromosome to form this prophage. Okay? And this prophage can replicate with the bacteria. But somehow, at some point, there might be some factors involved, maybe from outside, maybe due to other reasons, nutrients or environmental factors, which can induce this prophage to leave the bacterial chromosome. So this is what happened when the phage DNA leaves the chromosome here. You can see that it carries part of the bacterial genome, the bacterial chromosome, the red part, right, to form the phage DNA. And then new phage particles will be formed here. And in each phage particle, in each virus, it will carry a little bit of the donor DNA, donor cell DNA, or the host cell DNA. And then lytic cycle, the bacteria is lysed and the viral particle is released. And then one of the viral particle will infect another cell, and that's one we call recipient cell. And again, prophage is formed in the recipient cell. And it carries the donor DNA, the red part, into the recipient chromosome after the formation of prophage. So it carries this fragment from the donor cell to the recipient cell. Because this is mediated by the prophage. So usually the DNA from the donor cell or the fragment from the donor cell is the fragment adjacent or next to the prophage transferred into the recipient cell. So that is why we call it specialized transduction. While other parts of the bacterial chromosome cannot be transferred. Okay? So this is specialized transduction. I'd like to show you this video, this animation on this procedure.
Specialized transduction involves the transfer of only a few specific genes from one bacterial cell to another by means of a phage. The lambda phage, which infects E. coli, is a well-studied example of a specialized transducing phage. When lambda phage infects E. coli, the phage DNA enters the cell and then integrates into a specific site on the host chromosome. When an E. coli culture carrying the lambda phage is induced, phage particles are produced. On rare occasions, a piece of bacterial DNA, for example the gal gene, near the specific site of insertion remains attached to the phage DNA, and a piece of phage DNA is left behind. The phage that develop are defective because they do not carry the entire phage genome, but can still infect other cells. The defective phage can attach to another bacterial cell and the DNA can be injected. Both phage and bacterial DNA now become integrated into the new host chromosome. Only bacterial genes located near the site of integration of the phage DNA can be transduced, hence the term specialized transduction.
Okay, so this is specialized transduction. Okay. I do have another video showing us a summary of transformation, conjugation, transposition, and transduction. I think we still have time. I'd like to show you this video before we continue our lecture.
Welcome back. In this video, we'll be discussing the bacterial genetics, and we'll talk about transformation, transposition, transduction, and conjugation. We'll learn their meanings and how to differentiate between them. So first, let's talk about transposition. The bacteria has its DNA in two parts, either the bacterial chromosome or the bacterial plasmid. The bacterial chromosome is much larger and more complex and contains most of the bacterial DNA. And the bacterial plasmid is a small part of the bacterial DNA that has left the chromosome and now is floating inside the bacteria. So transposition refers to the process of extracting a small part of the bacterial chromosome to become plasmid, or reintegrating the plasmid into the bacterial chromosome. Keep in mind that if the bacteria want to share its DNA with another bacteria, it can only share its plasmid, not the chromosomes. So the plasmid is smaller and readily accessible. Just remember that transposition has to do with plasmid. Now let's talk about conjugation. So some bacteria have a sex balance, and we call these bacteria F positive, and other bacteria don't have this sex balance, and we call this F negative. Keep in mind that the sex balance is encoded by the bacterial plasmid, not the bacterial chromosome. So the F positive bacteria will link with the F negative bacteria and form the mating bridge. Then the F positive bacteria will make a copy of its plasmid, which encodes for the bacterial sex balance, and will transfer this copy to the other bacteria. So now both bacteria have a plasmid which encodes for a sex balance, and both are called F positive. This is the process of conjugation. Transformation, which is also known as competence, is the ability of the bacteria to take naked genomes or naked DNA from the environment and use this DNA. So for example, if we have some medium with some bacteria in it and we put some raw naked DNA which encodes for antibiotic resistance into this medium as well, this bacteria will be able to take this DNA from the environment and use it, and it will become antibiotic resistance. However, if we add a DNA lysing agent into the environment, it will degrade the DNA and the bacteria will not be able to use it. We see this exclusively in Streptococcus pneumonia, H. influenza, and Neisseria. So remember, transformation for fresh DNA. And finally, transduction refers to the process where a bacteriophage, which is a bacterial virus carrying its own DNA, will inject this DNA into the bacteria. This viral DNA will be integrated into the bacterial DNA and will start replicating the virus. If this results in a bacterial death, we call this a lytic phage. And if it didn't, we call this a lysogenic phage. If the bacteriophage did not kill the bacteria, aka a lysogenic phage, it will give the bacteria some superpower. So the bacteria now can produce exotoxins. Alright guys, that's everything for today. I tried to explain everything as much as I could. Hopefully I made this easier for you, and see you guys later.
Okay, so so basically it's a similar thing as we mentioned just now. And let's continue our lecture with the following topic. The next step, what we are discussing here, what we are introducing here are not in your textbook. It's not, probably it's not in any textbook, because these are the pretty recent discovery in research papers. So we'll introduce another mechanism called type 4 pili, which can also mediate DNA transfer. And previously we learned two types of pili, the common pili, which is useful for the bacterial adherence. And we also learned the sex pili, which is used in conjugation. But actually there are other types of pili which extend from the cell to the outside. And type 4 pili is another type of pili, which can grow from the bacteria, from inside to the outside, get long, and also it can retract. So during this extension and retract, it can pull the bacteria to move. And this movement we call switching. Okay, let's have a short video, and it will show us how the bacteria swim along the surface and how the type 4 pili work. Okay, let's see this video.
As seen in this movie recorded with a light microscope, Myxococcus xanthus cells glide forward in a mode that has been called social motility. Social motility is driven by a molecular machine called the type 4 pilus, which projects forward, attaches to a cell or surface, and then retracts. Type 4 pili can be visualized directly in 3D in a near-native state through electron cryotomography. Basal bodies without pili can also be seen. Aligning and averaging many of such 3D images results in a clear picture of the machine. Using diverse information about components, including atomic models where available, a pseudo-atomic model of the assembly can be proposed. Our current working hypothesis is that in the cytoplasm, the AAA ATPase pill B binds to the base of the machine. Conformational changes in the pill M ring likely propagate to the pill N, pill O cage, allowing pill A subunits to enter the assembly chamber. Hydrolysis of ATP drives cyclical conformational changes, which have been proposed to rotate a screw-like protein called pill C. Our new structural data strongly support this proposal. As pill A subunits diffuse into the basal body, pill C facilitates their incorporation into the pilus, allowing their hydrophobic tails to slip up out of the membrane into position within the growing stem. The hydrophobic tails of pill A subunits pack together to form the core of the strong helical polymer that occupies the axis of the basal body, again visible by electron microscopy. Pili can grow to several microns long in seconds. After the pilus tip attaches to a surface, a tension-induced conformational change likely signals structural changes in the basal body. These changes release pill B and creates a binding site for pill T, the AAA ATPase that drives retraction. Pill T likely rotates pill C in the opposite direction, slipping pill A subunits back into the membrane. Cycles of pilus extension and retraction move cells forward.
You can see that bacteria are single cellular organisms, but with the help of type 4 pili, they can form groups and glide on the surface in in lines or in groups. And so this is the primary role of type 4 pili. It can help the bacteria to move. But the type 4 pili have another property, that when the pili is extended from inside to the outside, it can grab the surface and pull the bacteria. It can also grab DNA molecules. So there is some affinity between DNA and this pili, so that this pili can grab some DNA molecule and pull the DNA molecule back into its cellular body, so that it can help the gene transfer by actively transport DNA into the the bacterial cell. So this is what happened in the type 4 pili for DNA uptake. To study this procedure, to study this hypothesis, the author created a special strain of Vibrio chlora, so that this bacteria can express a type 4 pili, this green fiber. They can stain the type 4 pili with green fluorescence, and they can image the live bacteria. This is a live bacteria, not fixed, not killed. So that the bacteria can extend the type 4 pili outside, grab some DNA, and take it back. See here, this red color is a DNA molecule stained with red fluorescence. So it can take DNA and then draw it back into the cellular body. And this is the cell, the Vibrio. You can see the typical shape or morphology. And the cellular body is stained in green color, green fluorescence, and the DNA is in red color. So if you mix red and green color together, you will have this yellowish spot, yellow color. So this spot means that inside the bacterial body, there is some accumulation of DNA at this spot. So the bacteria take up DNA and accumulate at this spot. So this is a very interesting procedure that the bacteria can utilize type 4 pili to initiate DNA uptake. And still, there might be other questions you want to know, because the DNA is accumulated here. Is it inside the cell, or is it in the periplasmic area space? That I think is not clear yet. But according to my work, is that if the bacteria cannot produce type 4 pili, the uptake of DNA will be very difficult. So type 4 pili actually help the bacteria to take up DNA, to take up plasmid. So this is a very interesting topic and a very interesting research work, and we can follow up. Okay, so after learning this lecture, we should learn some basic terms, and we should understand some basic terms, including genetics. Genetics defines and analyzes heredity or constancy and change in a vast array of physiologic functions that form the properties of organism. So this is like any heredity or constancy or change. We are talking about the variation in the DNA level or genetic material level. And we should also understand phenotype. Phenotype is the observable physical or biochemical characteristics of an organism. And I mean, in the microbiology part is the microorganisms. So this phenotype, you can observe it, you can detect it, like whether it can produce something or degrade something, whether it can move or not. All these physical biochemical properties can be defined as phenotype. While genotype means the genetic makeup of a cell or an organism. Genetic makeup means what are their genetic material, what are their DNA. So that's the genotype. There are some change or mutations which can be divided into two groups. One is a genotypic mutation. Genotypic mutation means that this change or this variation happens on DNA level, on its genetic material. And because it happens on the DNA molecule, this change can be inherited, can be passed from one generation to another. So it can be passed from one cell to its daughter cells. This is genotypic mutation. Another change or another variation we call phenotypic variation. Phenotypic variation means that the phenotype changed. So the phenotype is different from previous state. And this happens because of the influence of other factors, but not on its DNA level. Maybe due to the change of temperature, maybe due to the change of the environmental osmotic pressure, or maybe because of the change of the nutrients or nutrition condition. So because there is a low level of nutrients or high level of nutrients, the phenotype changed. So this we call phenotypic variation. It doesn't happen on DNA level. This we call that this variation is reversible. Reversible means that if the environmental factor changed back, the phenotype will also change back. It's not heritable. It cannot pass to its daughter cells. So this is several terms we need to understand after we learn this topic. Okay, so I think basically that's what we are talking about for this large topic, the bacterial genetics and variation. And we covered the major points in this area. And for the next test, some of the questions will be designed on this topic, and some of the questions will be designed according to what we learned previously, including disinfection, sterilization, and maybe something we learned before. So you guys can spend some time to go over our material, to read our textbook, and then prepare or get ready for the test. The test will be open for 3 days. So you can take your time, and for your own convenience, you can just spend maybe half an hour to finish it. Once you started the test, you'll have half an hour to finish the 20 questions. Okay.